Good morning, everybody. I am Andrew Buddymeyer. I was diagnosed at the age of 15, and I'm here with my family, my mom, my dad, and my 3 younger brothers. I'm the only affected individual in my immediate family. I also have a cousin who has Barth syndrome. Right now, I'm about to graduate from college. I graduated in a week from Florida State University, and thank you. And then I plan on going to law school in a year, and we're also in Tampa getting ready to kick off our 5th annual golf outing benefiting the Barth Syndrome Foundation, and we've raised over $100,000 since we started these golf outings. And now I get the great honor of introducing Dr. Richard Kelley, who, I mean, some of you may know I went through a little period, I guess, the last couple of months, and he was a great asset to our family, and he was able to get on the phone and basically completely change our doctor's demeanor and his outlook on everything. I mean, he went from kind of, we're not in a great situation to, we don't even need to talk about, this transplant stuff anymore. All we need to do is get you to eat, He also took a lot of my blood for a while, but so Dr. Kelley is a professor of pediatrics at Johns Hopkins University School of Medicine, as well as director of the Division of Metabolism and a staff physician at the Kennedy Krieger Institute in Baltimore. He is the chairman of the International Scientific and Medical Advisory Board of the BSF and the co-recipient of the 2008 Varner Award from the BSF. Dr. Kelley specializes in the diagnosis and treatment of inborn errors of metabolism. His research focuses on the determination of the biochemical basis of both known and novel genetic disorders and on the treatment of selected diseases, including Barth syndrome, Smith-Lemli-Opitz syndrome, and disorders of mitochondrial metabolism. Dr. Kelley also is a co-founder of and consulting geneticist for the Clinical for Special Children, a charitable medical facility for the diagnosis and treatment of genetic disorders among the Amish and Mennonite peoples of Lancaster, Pennsylvania. Dr. Kelley's topic is nutrition and metabolical aspects of Barth syndrome. BSF is very fortunate to have been able to rely on Dr. Kelley over the years, and we are honored to have him speak with us today.
So this is what I'll be talking about, and I would like to get to questions as quickly as possible. I think that's where we're going to learn the most. But a lot has happened in the past 2 years since the conference here in 2008, where we had the opportunity in a fairly systematic way. Oh, I see. I'm sorry. Okay. I didn't know I was going to get written questions. We had the opportunity to get blood studies for the first time in a very consistent manner from a large number of boys, looking at a lot of different biochemical parameters. And we know we know where the primary biochemical problem is in cardiolipin, but the consequences of that in the whole body are quite extraordinary, at least the number of systems that are affected. So we made sort of a one fell swoop attempt at getting together as much information as we can. And the important thing about doing it here last time was doing it in a carefully timed manner, because most metabolic changes in the body are dependent on what someone eats. So when you normally go to a doctor's office, you're munching on a bag of chips or doing something, and you really can't interpret studies done that way. We got very good data from a scientific standpoint. We got excellent data. And we learned an awful lot from that. We had suspected from a long time ago that there was a significant amino acid abnormality, and there are a lot of features of Barth syndrome which can be explained on the basis of an inability to make muscle protein, including heart muscle protein. And it was mysterious for years why the particular abnormality in Barth and cardiolipin would lead to the biochemical abnormalities that we see. And suffice to say, I treat a lot of different mitochondrial problems, and we just do not see Barth syndrome type problems in most of those. So I've always thought of Barth syndrome as not a regular mitochondrial disease, and therefore it requires a new approach, as you all perfectly well understand. So what I wanted to show today is quickly go through some of the things that we found, what we think it means, and how it has changed our thinking, and how it should change what you should be doing. So, make this thing work. So these are the questions I would like to try to answer today, and then I have all your own contributions here. So, why does the cardiolipin deficiency cause Barth syndrome? That was not clear, still a little unclear, but at least I have some strong biases right now. Is cardiolipin the only abnormality in Barth syndrome? Short answer, no, and I'll show you why. And that's very important. That's been the major change in my thinking in the past year. What do we have to do to fix the Barth biochemistry? We're learning. Andrew has contributed his blood and his wisdom to figuring out how to do that. And based on those kind of little experiments that we've done, what should the Barth diet be? We're still working on that, but I can give you some basic ideas about it. Real major problem that many of you have seen and we heard about this week is how much should the Barth infant be fed? And I say infant because the infant has very little say in what he's going to eat. And therefore, and when a child is underweight, various directives are given about what a child should have, and they're often very far off what they should be, and I'll try to explain why. Another important question that comes up a lot is, does growth hormone work in Barth syndrome? And the short answer to that, again, is no, and that may seem a surprise to some people, but I'll explain why. And what is the cause of diarrhea? We have an inkling of an idea of what's causing the diarrhea. So what will cause a real sea change in our thinking about Barth syndrome was the fact that there were two types of Barth syndrome. Now, syndrome, to a geneticist like me, I trained as what's called a dysmorphologist who studies syndromes. And back when I started, we didn't know hardly any genes. We just had to say, this child looked like this syndrome, this child looked like that syndrome. So as a syndromologist, I had to recognize that there were two types of Barth syndrome, even though we all think of the X-linked form as Barth syndrome. But there really are two forms. And I just listed here what the differences are, and there are almost no differences. They have essentially all the same features. The main difference being that intelligence is quite normal in Barth syndrome, and it is not very abnormal, but they can have a lot of trouble with brain seizures and developmental delay, and we think we can treat that separately, but that's the major distinction. The other thing that stands apart from any other metabolic or genetic syndrome I know is that Barth syndrome has this extraordinary pubertal growth. They're very retarded in growth often until they get to be 13, 14, 15, and then zoom. They have to do all their pubertal growth, or they have to do twice the normal pubertal growth in the same period of time, and that's very debilitating, and it leads to heart failure in children. And this other syndrome here, which is a recessive syndrome, which means it affects both girls and boys, it's an autosomal recessive. They have exactly the same growth pattern. So here you have all these features that are essentially the same except for one, and the same growth pattern. As far as I'm concerned, that's biochemically the same syndrome, or they're working on there's an abnormality in the same system. And so we strove to figure out what the abnormality was in this other syndrome. We knew that cardiolipin was deficient in Barth syndrome, but we didn't understand why it was causing the problems it caused. It didn't help us understand Barth syndrome, knowing that it was cardiolipin deficiency. So we figured if we could determine the cause of this other Barth syndrome, then we might put two and two together and say, aha, this is the common feature. And, oh boy, this doesn't turn out. But what we found, and another group in Canada found virtually at the same time, was that in the recessive form, the autosomal recessive form, it's an abnormality. It's this protein right here. One of these two proteins here is deficient in this other syndrome. And this is what's called a mitochondrial protein import motor. So it's a complex of proteins that pulls enzymes into mitochondria. Most enzymes in mitochondria are made outside the mitochondria and have to get in to do their work. And specifically, this import motor here, there are two types of import, actually three types of import motors, but this system here is specifically built to bring in the enzymes that work inside the mitochondria. The energy-making part, the ATP synthesis, occurs in this membrane right here. But a lot of the conversion of food into substances that the energy-generating system can make come from what's called the mitochondrial mixture, the mitochondrial matrix, the inside of the mitochondrion. So we have one disorder where there's cardiolipin deficiency, and cardiolipin, as you all know now, is the major phospholipid, the major structural lipid in this membrane, and an abnormality of this protein here. Biochemically, molecularly, the conclusion is obvious that cardiolipin, cardiolipin in these children, completely normal. So that was our first puzzle. Here you have the same syndrome that should be caused by cardiolipin deficiency, and the cardiolipin is normal. So the way to put that together is to say that in this syndrome, the protein motor there's a primary mutation in the protein motor. In Barth syndrome, X-linked Barth syndrome, the problem is that this system, which requires cardiolipin, is the most sensitive to the cardiolipin deficiency. So we can measure all sorts of little deficiencies in the mitochondria in Barth syndrome, but what really hurts, what really affects the cell, it appears, is the deficiency in this import motor. So that, and that has gone on to be demonstrated in the laboratory. That kind of work has not been published yet, but it's going to be. So the idea that a major consequence of cardiolipin deficiency is failure of protein import is now, at least in my mind, established, and that is our operating diagnosis, so to speak. This is our operating model, how we're going to go forward in understanding how to treat Barth syndrome. So what does that import motor do? Well, let me show you first. I'll go back here. So what this is the citric acid cycle. And the citric acid cycle is truly a cycle. It's a group of enzymes, and here are the enzyme names. And they convert one compound into the other. And a lot of energy in food comes in through here to make citrate. And it's called the citric acid cycle. And then citrate is broken down, and in the process of being broken down, energy is made, and then it comes back here and is regenerated from this compound, acetyl-CoA, to make more citrate, and then you go through the cycle again and make more energy. All of these enzymes require that import motor. And when there is something like a lipid abnormality that affects a protein complex, usually the protein complex still works, but doesn't work quite as well. And we had noticed 25 years ago that boys with Barth syndrome had an increase in this compound right here, aconitate. Not very high, but it was increased consistently. And this is one of those citric acid cycle intermediates. We also see here the methylglutaconic acid, which is diagnostically very helpful, but probably means very little from a nutritional metabolic standpoint. So what does that mean? What that means, with my interpretation, there's a relative block at aconitase. It is still there and functioning, but there is a relative block. A relative block here causes a deficiency down here. And the cells, for a cell to survive, it must keep this cycle going. It must be able to make all of the components in this cycle. So how do you do that? If you can't make alpha-ketoglutarate from citrate in the normal way, what does the cell do? It very logically, but to the harm of the body overall, it pulls in these amino acids, and in particular arginine, that can be turned into alpha-ketoglutarate. So what is happening is this system is driven so far that the arginine level becomes too low to support protein synthesis. So to keep the cell going, it reaches that very minimum of arginine level that will allow the citric acid cycle to keep going and yet still allow a little bit of growth. And and that will, so that, and the consequence of that, what we see from that from a dietary standpoint, is that many of the boys will eat a lot of protein, yet they still have an arginine level that is at the lower limit of normal. And when we measured the levels in 2008, every boy had almost exactly the same very low arginine level. I can show you that. Wrong way. So what this is a graph showing the relative levels of amino acids in the blood. I've just chosen 5 here. But this is the normal range, this blue bar. And this box is what we found in the Barth syndrome children. And so cysteine here is a little bit low, but all the other ones are basically normal. So this overlaps the normal range, this overlaps the normal range. This is a no-brainer. That's not normal. It's really, really low. And it's too low. Can you give me a Kleenex? There's something I'm allergic to up here. So this is really low, and this is too low to support protein synthesis. And this protein here that we measure, prealbumin, is a general marker of the rapidity of protein synthesis, and that is deficient as well. So, and this we had seen low arginine levels, but we see low arginine levels in other conditions, not very often, but it was the absolute consistency. We can look at the amino acid profile overall in Barth syndrome and literally make the diagnosis of Barth syndrome from looking at the amino acids alone. There are other abnormalities, something high, something a bit low, and it fits perfectly. And what was extremely gratifying, so to speak, or consistent with this hypothesis, so again, to summarize, these amino acids are being pulled in here, and these amino acids are being pulled in there. So the process of metabolizing these compounds is increased. And the way this is done is an enzyme is turned on called arginase, which breaks down arginine. And basically, as soon as a child eats arginine, it gets in the bloodstream, it's broken down to ornithine, which then goes to alpha-ketoglutarate. So they now have a mouse model of this, and they studied the muscle in the Barth mice. And what did they find? The oxidation of glutamate is about double, about twice normal. So that more or less confirms that and succinate too. These two parts, these are the two different ways that one can get amino acids in at that level. The rate of metabolism at those two steps is increased. So it doesn't prove this hypothesis, but it fits very nicely. And basically was the only significant biochemical abnormality they found in the Barth mice, in the mitochondria of the Barth mice. They looked at a variety of other things. And as we know in Barth syndrome, the synthesis of energy, ATP, is normal. So the cell goes through all of these extremes of biochemistry simply to maintain ATP synthesis. And we've known that for 25 years, that ATP synthesis itself is normal. But that is produced at a great cost to the nutritional status of the body. Oh, I can't go backwards. So, what does one do? I should say we first focused on treating arginine, but what turned out was that this enzyme that is turned on, arginase, is so active that we could give 20 grams of arginine a day and couldn't get the arginine level all that high. Yes, it would go up, but it would be, clearly the cell is still spinning wheels. And so then I focused on cysteine, and I went to cysteine partly because it was a little bit low, but partly because it has a separate function in the metabolism of the cell to tend to tell the cell to slow down this process of breaking down protein. So what is happening is if the protein is not in the diet, the protein from muscle is broken down and turned into arginine and cysteine and these other things. So, but cysteine, whereas arginine by itself can't regulate this process, cysteine, high levels of cysteine, or at least normal levels of cysteine, suppress this process to a degree. And that's indeed what we found in the few boys who've been given cysteine after starting arginine. It has reduced the amount of arginine that is needed to be given. The arginine level has gone up. And it has, I'm sure many of you know Matt Galley, got an email from his dietitian on Monday, and he has gone from eating, he's now on both, he has gone from eating 600 calories a day in January to 1,200 calories a day in March, and now he's eating 2,500 calories a day, partly because to normalize the amino acid biochemistry improves appetite. And he has said that the cysteine really makes him hungry, and we've heard that from a couple of the other children. So we're still trying to work out the details. The long-term goal is to prepare a special Barth supplement to be able to give to children. And giving, as Andrew will testify, taking arginine sometimes has its problems. And it's salty, and if it's swallowed in capsule, it turns out it can get stuck going down. So it's not perfectly, we haven't worked out the best way of doing it. But in general, it works fairly well. But the long-term goal is to put all the needs together and as a single supplement, have that produced by a pharmaceutical company or formula company, and then that'll be the Barth supplement. The other thing I should say, because amino acids are utilized so rapidly, they there's an increased need for B vitamins. So we've also recommended that the children start taking, especially pyridoxine B6, but there's probably a need for other B vitamins. So many children are taking so-called B50 tablets or B complex. It's pretty benign to do that. And we found in a couple of boys who were on intravenous feedings, where they use a lot of extra methionine in place of cysteine, we found they couldn't convert methionine into cysteine. And by giving them extra B6, pyridoxine, they could do it. So we knew they were sort of borderline deficient pyridoxine. So how does one do this in a practical sense? Well, one can look at, I did this a couple of years ago, one can look at the arginine content of foods, and there are certain foods that are very high in arginine. And for example, the highest one, so what I've calculated here, I should say, is here's the arginine per 100 grams of food, but you see some of the foods are nuts, which are high in fat. So it's a good source, good packaged source of arginine, but per calorie it's not as high as, for example, lentils, just about any kind of bean, a legume. Of the meats, chicken is high, and eggs are the highest. So one can look at this list and see that certain foods are not especially high in arginine. Whole grain wheat flour is not as high in arginine, happens to be as high in cysteine. So wheat bran at least can be may be part of the diet. And if this is courtesy of Sonia Schlepak, who's done an awful lot of the analysis here on the cysteine side of things, and she has calculated out the same kind of information. I don't have the data here, but foods that have a high cysteine content, and that's cysteine per calorie. So you have to think of it on a per calorie basis. Just looking at the number of milligrams of cysteine in a certain food may mislead you if it's a low calorie food. So one wants to take a number of foods that are high in cysteine. And it is, I'm sorry these don't show up well. And again, this is how, so one of the questions is, can you do this without taking powders and pills? And the answer may be yes. And Leo, the boy who has benefited from the cysteine and arginine analysis, is getting all of his arginine and cysteine by emphasizing foods that are high in arginine and cysteine. This is an example of the meals that he gets. And I'm sorry that it doesn't show up as well. They look really good when you have them on a good screen. And it's remarkable. We see the amino acid abnormalities showing up by the end of the first year. It's interesting, and it's another whole story about what's going on with the Barth heart, but the amino acid abnormality does not show up until after the first year in most cases. Even though there's heart failure, I now think there are two different causes of heart failure, Barth syndrome. One is amino acid and one is fatty acid. But suffice to say that by Leo's age, the amino acids are usually abnormal, and his amino acids are completely normal. So he's on no artificial supplements, just what any dietitian would say is an excellent diet. And so what we're doing right now is we're working on trying to put together, there are some other issues about energy requirements. I have one slide about that. So there are a number of goals to achieve to get the right amount of calories, not too many, not too few, right amount of arginine, right amount of cysteine. There'll probably be other nutrients, but we've put together a small team. I wanted Nicole Clayton, just to stand up, who's been with me. She sat through a repetition of the same speech, probably 30 times in the past two days. So she knows the mantra quite well about the nutritional aspects of that. But she's a really good dietitian who thinks analytically, can analyze energy requirements, and we're going to work together to put together a document that'll say this is what the ideal Barth diet should be. Likely it'll change in years to come, but right now I feel that we have enough information to make these kind of recommendations. But I'm very encouraged to see that by eating food that's really good for you, I suspect that you might be able to treat this. And I have a sense, based on some other anecdotal experiences, that if Barth syndrome is treated well very early, it's always easier to treat. Once you get into a hole, it's hard to get out of it. This is the garden that Leo has, where some of Leo's food is grown, and here's Leo selecting his breakfast. And you can see he really likes it. So, the other thing I just want to mention is we had the opportunity last time, 2008, to measure lipids. So, cardiolipin is a phospholipid. A phospholipid has lots of fatty acids in it. And this is a diagram from some work that Michael Schlama had done on some cells I supplied him. And he looked at mitochondria and found that, you don't have to look at all the numbers, but there were differences between control and Barth syndrome in the mitochondria, but there were also differences seen in what are called the microsomes, which are closely related to the mitochondria. So it wasn't clear whether the abnormality was sort of leaking over or affecting the microsomes, or whether the microsomes themselves had an abnormality, because cardiolipin, or TAZ, is supposed to work only in the mitochondrion. But other abnormalities in Barth syndrome are very difficult to explain on the basis of mitochondrial abnormality. And so we looked at the lipids, and this is a diagram showing what we found in the red cells of boys with Barth syndrome. Red cells have no mitochondria. They last for 6 months or so. They have their own phospholipid generating systems. And what this shows is that and this is the same pattern you'd expect if TAZ were not working. High saturated fatty acids, low levels of certain unsaturated fatty acids. This is one of the fish oils as it happens, and low levels of things like oleic acid. These are standard deviations that normal is between -2 and 2. So you can see here that overall it's completely different pattern from normal. I've never seen anything like this, and certain ones are quite clear. And this is the average of about 24 boys. Some boys were -4 or 5 or -5 standard deviations on some of them. So there's a severe abnormality in the lipid membrane of probably all cells have an abnormality in lipid membrane. And that is important because hormones interact with lipid membranes. So now we're beginning to think that even the neutropenia may be a hormone receptor problem, that the GCSF is not interacting normally. And there's precedent for that in other metabolic disorders where a lipid abnormality interferes with the function of hormones.